How's everybody doing today? It's a nice kind of gloomy day, maybe a little drippy. Um, Anybody ready for some sunshine? I don't know, maybe. Hopefully we'll get a little bit. I did get out for a walk yesterday. I got a little wet, but surprisingly, I did not melt. So I guess I'm not made mostly of sugar, but you guys already know that. So this is not a surprise. Right, let's see. Before we jump into new stuff, I just want to remind you that we have an exam next Friday. I don't remember who mentioned it at the end of class on Wednesday, but I thank you for the reminder. And so we will try to get through photosynthesis today and give you guys a good understanding of photosynthesis. And then next week, we'll touch a little bit on plant nutrition. So the main way, I hope you'll understand by the end of this week, by the end of today, that the main way that plants get food is by doing photosynthesis and making their own food out of the carbon dioxide in the air. But plants do need mineral nutrients to stay healthy. And so we'll talk about that a little bit next week, and that will all be on your exam. And then hopefully most of the last quarter of the semester we can spend on kind of the biodiversity part of our course. So getting out into the kind of ecological level. So that's kind of where we're going. So let's see, we are working on the light reactions of photosynthesis. So we've already looked at the basic properties of light, how light behaves as both waves and particles. And we looked at how pigments absorb light. They bump electrons up to a higher energy level. And then we were working on the light reactions, or we were just getting ready to work on the light reactions when we left off last time. So just to review, I'm going to show you a couple slides that we already looked at. So photosynthesis, as you know, occurs in the green parts of a plant, which are predominantly leaves. This is a perfect time of the year to study photosynthesis because as I look out my window, my beautiful window in my guest bedroom, I can see leaves starting to grow on the trees. This is a very exciting time of the year for a plant biologist. Maybe some of you not so much, but another exciting thing is that we're getting toward the end of the semester. So that's also exciting. Anyway, it's spring, the plants are growing, they are producing leaves. And so leaves are what keeps the plant alive. Leaves are what produce the food of a plant. So that process of photosynthesis happens in chloroplasts. And a chloroplast, as you know, has these structures. It's got these thylakoid membranes on the inside, and that is where the chlorophyll molecules are embedded in these thylakoid membranes. And the thylakoid, and so remember the process of photosynthesis has both the light reactions and the dark reactions, and the light reactions happen in the thylakoid membranes. The Calvin cycle, which we will get to in a few minutes, the Calvin cycle happens in the stroma, which is the kind of liquid part outside of the thylakoids. But you will remember that in the thylakoids, let's see. Oh, let's go to slide 33 is where I want to go. In the thylakoids is where the light is absorbed. So the light reactions involve the absorbance of light, and mostly in the red and blue wavelengths, as you will remember. And so how does a pigment absorb light? we learned that the pigments, when the pigment absorbs light, that means that an electron is bumped up to an excited state, to a higher energy level in the atom, and then it immediately falls back down. And that chlorophylls are pigments that are embedded in photosystems. And so here is a diagram of a photosystem, and it shows all of these pigment molecules. Once a pigment molecule receives a photon, an electron is excited. And then when that electron falls back to its ground state, it releases another photon, which is absorbed by another pigment. And that causes another electron to be excited. And when that electron falls back to its ground state, another photon is emitted. And so photons keep getting passed from pigment to pigment until finally a photon with just the right amount of energy is released, and it is absorbed by these chlorophyll A molecules in the reaction center of the photosystem. And an electron is not only excited, but it is released from these chlorophyll A molecules and captured by the primary electron acceptor. And so this is really the first step in electron transport. I had to mute myself there for a second so I could cough, but I'm back. Okay, so this is the first step in the electron transport, is this electron getting ejected from these chlorophyll A molecules in the reaction center, and that electron being accepted by the primary electron acceptor in the reaction center complex. So this is a photosystem. And I've kind of explained to you what this figure illustrates. And I already gave away this answer, that this electron moving here, this arrow showing the electron being donated to the primary electron acceptor, that's the first step really in the light reactions. So here's my next question. I just showed you a photosystem. What's the difference between photosystem 1 and photosystem 2? So this is the first question that I would like to have you answer in the private chat. So send me, I have to go back to this because I gave you guys the wrong answer on this multiple choice. The correct answer actually is A. Photosynthesis 1, or PS1, photosystem 1 was discovered before PS2. That is the correct answer. B is incorrect. It's actually PS2 that receives light first. And PS2 has a wavelength of 680. And so, so answer C switches the wavelengths, and answer B is incorrect. So the correct answer to this is answer A. I'm sorry, I gave you the wrong answer. So you were working on this task to model the linear flow of electrons. The thing that almost everybody did not do was show how the flow of electrons generates a proton motive force, which then is how ATP is produced by chemiosmosis. So most of you guys showed what happens with the electrons, and you showed me generically where the ATP comes from, but you did not show me the mechanism of how the ATP is produced. So let's look at it. Most of you drew what we call the Z scheme. And do you see why it's called the Z scheme? Do you see how the electrons kind of go in a Z in this diagram, or as kind of a zigzag? So this diagram, it does not show energy, but on this figure, energy is sort of on the y-axis. So it kind of shows the free energy levels. And I might just draw that in so you can see it. We'll draw it with red, and I'll just draw an arrow like this. And this is free energy. Free. Okay, I'm going to switch to cursive because that works better when writing with a mouse. So this figure shows free energy. And so let me just walk through it with you guys in case you were able to draw it but don't really understand it. Let me see if I can help you understand it. So as we talked about last time, the light harvesting complex, pigments in the light harvesting complex will absorb light, and then the light energy gets passed from pigment to pigment until finally we have the chlorophyll A in the reaction center that is absorbing just the right amount of energy in a photon. And that can move an electron from the chlorophyll molecule up to the primary electron acceptor. Now, this leaves a hole of electrons. So electrons have left the P680 and gone up to the primary electron acceptor. And so in order for another electron to be passed up, this P680 has to receive electrons, and those electrons come from water. So the source of energy is the light. The source of the electrons is the water. So water is split and electrons are moved from the water to the P680. This P680 is highly electronegative once it loses this electron. As the water is split, this releases hydrogens and oxygens. The oxygen immediately combines with another oxygen to make O2, and it diffuses out of the chloroplast and out of the plant cell and out the stomata and into the air. This is the source of oxygen for us to breathe. The hydrogens are retained inside the thylakoid space. Meanwhile, the electrons are passing through several electron carriers. And similarly to what happens in the mitochondrial membrane, as electrons are passed from the primary acceptor to the plastoquinone, and from the plastoquinone to the cytochrome, and from the cytochrome to the plastocyanin, and hydrogens, and I'm going to have to draw this in also, hydrogens, oh, I should have left my pointer. So here we are back with our pointer. I will draw, yeah, I'll do this in red also so we can see it. Hydrogens that are actually outside of the thylakoid membranes, they get moved into the inside of the thylakoid membranes. So just like in the mitochondrion, the electron transport pumps hydrogens, and that creates an electron motive force, which I will show on the next slide. And it is that stored energy of the hydrogen ions that is used to make the ATP. So this shows an arrow coming to ATP, but I want you to understand that the ATP does not come directly from this electron transport chain. And we'll get to that in a minute. Okay, so the photosystem 1 now, which has P700s at its center, the P700 here accepts an electron, and it requires another photon of light energy to boost that electron to the second primary electron acceptor, which then passes the electron to ferredoxin. And finally, the electron ends up reducing NADP plus. And so the final electron acceptor is the NADP plus, and those electrons end up being stored in NADPH. So electrons that started in water flow through 2 electron transport chains and finally end up in NADPH. And as the electron goes through these 2 electron chains, these electrons lose energy, as you can see represented by their level on this figure. And that energy is used to pump hydrogen ions, and that proton motive force is then used to make ATP. So let's move to the next figure. And this figure from your textbook shows the same light capture, the same electron movement through these 2 electron transport chains, but this figure shows the locations more like they actually are in the thylakoid membranes, and it also shows the pumping of the hydrogens. So a hydrogen ion concentration develops so that the inside of the thylakoids has a higher amount of hydrogen ions than the outside because of the pumping of the hydrogens as electrons travel through these electron transport chains. And then these hydrogens flow through an ATP synthase molecule, and this is what actually generates the ATP. So this is chemiosmosis, very similar to what happens in the mitochondria. So now I hope you can see that the ATPs do not come directly from the electron transport chain, but instead the ATPs are formed when hydrogen ions diffuse through the ATP synthase. So this is chemiosmosis, and let me write that in, chemiosmosis. And somebody wants to be let in. Participants. All right. So what questions do you guys have about this? This is all kind of complicated. So what questions do you have? You can send me a totally understand it. Does anybody have a question that they can understand? Okay, so somebody said, maybe I could just do an overview of the whole process one more time. So I will do that. All right. So let's use this diagram to do an overview of the whole process. Remember, this is these are the light reactions. And I want to just remind you of the kind of the big picture is that the light reactions are going to take the light energy that's coming in, use that energy to make ATPs, also use that energy to cause electrons to come from water and be stored in NADPH. So the 2 main products of the light reactions are the ATP and the NADPH. So maybe it's helpful to take a step back and look at kind of the big picture. Light energy is transferred to ATPs. That energy is also used to move electrons from water to NADPH. And then the NADPH and the ATP are going to be used in the Calvin cycle to actually make sugar. How does this happen? The light is absorbed by chlorophyll. And as a result of the light being absorbed by these chlorophyll A's in the reaction center, an electron is boosted up to an electron acceptor. Then that electron is passed in a series of redox reactions through plastoquinin and cytochrome and plastocyanin until that electron arrives in photosystem 1 in another pair of chlorophyll A molecules. And another electron gets boosted up to an electron acceptor. Again, the energy comes from the light that is absorbed by this photosystem. So light is absorbed in two places, both by photosystem 2 and by photosystem 1. And that energy is used to boost an electron, first in photosystem 2 and then in photosystem 1. And finally, that electron goes through another short electron transport chain until it finally reduces NADP plus to NADPH. At the same time, this flow of electrons through this electron transport chain causes hydrogens to be pumped into the thylakoid membrane. So lots of hydrogens build up on the inside of the thylakoid in what we call the thylakoid space. So you see there's a high hydrogen ion concentration. A very high hydrogen ion concentration makes the inside of the thylakoids very acidic. And because of that high concentration of hydrogens inside the thylakoids, those hydrogens tend to flow down their concentration gradient. And the only place they can get out of the thylakoids is to go through an ATP synthase. This causes that molecule, a part of that molecule, the ATP synthase, to actually spin. And this provides energy to combine ADP and inorganic phosphate to make ATPs. So the final products are NADPH and ATP. All right, now show me thumbs up, thumbs down, thumbs in the middle. Was that at all helpful? I see a few more thumbs trending upward, if not all the way up there. Yeah, maybe trending upward. Okay, so what I recommend is that you guys, between now and the time you have your exam, a good way to understand what's going on is to ask yourself questions about it. Some of the questions I've asked you, like, where does the energy come from? Where does the light come from? What's the final electron acceptor? What causes the ATPs to form? Explain how the ATPs are formed and where that energy comes from. So ask yourself questions about it. Try to draw it out yourself on a whiteboard and see what you can remember, what is still fuzzy to you. Get with a partner and talk it over with your partner. You can go to science help sessions and talk it over with one of the tutors there and kind of just work on getting your understanding. All right, let's move on then to see how the ATP and NADPH are used in the next step. So let's answer this question first. And that will be another measurement of what you've learned so far about the light reactions. All right, so most of you have had a chance to get your answer in. A couple of you corrected your answers. The correct answer is water. The electrons come from the water, and somebody asked, well, where does the water come from? Well, think about it. How does a plant get water? It absorbs water from the soil, right? The soil has to go up through the stem of the plant and out to the leaves. And so the water is in the spaces on the inside of the leaf, and it gets there by coming up from the roots through the stem of the plants to the leaves. This is happening as we speak with your plants that are in the environmental chambers. Hopefully, if you've watered them. So let this be a reminder to go water your plants because they need the water in order to do photosynthesis, in order to produce the food they need in order to grow. All right. So let's look at how the ATP is used once it is made by the light reactions, and the ATP is used in the Calvin cycle. So your learning objectives for the Calvin cycle are to be able to model the Calvin cycle and identify the role of rubisco, which is arguably the most important enzyme in the world, and perhaps the most abundant enzyme in the world. Your plant leaf cells are full of rubisco. And we'll look at the three main steps of the Calvin cycle. And then hopefully by the end of our lesson today, you will be able to trace the energy of a photon from light, or a photon of light from the sun until it gets into a molecule of glucose, and trace the atoms of carbon from CO2 until they get into a molecule of glucose. All right, so let's get started with the Calvin cycle. All right, remember the big picture. We just looked at the light reactions. Light energy is stored in ATP, and electrons from water are stored in NADPH. The ATP and NADPH then provide the power, the reducing power and the energy to run the Calvin cycle. And the goal of the Calvin cycle is to incorporate inorganic carbon from carbon dioxide into organic molecules that would be the overall product of photosynthesis, the sugars that are produced by photosynthesis. These reactions of the Calvin cycle happen in the stroma. All right, so based on your reading, can you identify the main events of the three phases of the Calvin cycle? You can send your answer to your partner, your lab partner. Three main events of the three phases of the Calvin cycle. So I think you've had enough time to look at that. And I got correct answers from a couple people whose lab partners are not here. So let's look at the three phases. The first phase is carbon fixation. What does that mean? What that means is that a carbon dioxide molecule, an inorganic carbon, is added to an organic molecule, in this case, a 5 carbon molecule, 1, 2, 3, 4, 5, that is called ribulose bisphosphate. I want you to pay attention to the difference between ribulose bisphosphate, RUBP, and rubisco. Rubisco is the enzyme that facilitates the combination of the carbon dioxide and the RUBP. So if you have a 5 carbon molecule and you add on 1 carbon in the form of carbon dioxide, the molecule that results should have how many carbons? I'm not hearing any answers. You have 1 5 carbon molecule, you add on 1 carbon dioxide. So how many carbons does the resulting molecule have?
6.
6 is correct. Okay, so you can see them here, 1, 2, 3, 4, 5, 6. So this 6 carbon molecule. We don't know what this 6 carbon molecule is because it does not last long enough for us to isolate it, and it immediately dissociates into 2 3 carbon molecules called 3-phosphoglycerate. Now, you might be wondering why you're seeing 3s. So we usually represent the Calvin cycle in 3s because it just makes the math work out better. So if you have 3 of these 5 carbon RUBPs that combine with 3 CO2s, then you will get 6 phosphoglycerates. This is called carbon fixation because a carbon dioxide is fixed into an organic molecule. This carbon goes from being inorganic to being organic, to being combined with other carbons. So that is carbon fixation. The next step is where we use the NADPHs and the ATPs. And so this stage is called reduction because we're using electrons from NADPH to reduce the phosphoglycerate and rearrange it a little bit to get glyceraldehyde 3-phosphate, G3P. So we use ATP to phosphorylate it. So the phosphoglycerate has one phosphate on it. If we have 6 of these molecules and we use 6 ATPs, we can put another phosphate on the end of each one of these. So now we have bisphosphoglycerate, meaning there are 2 phosphates. That's where the bi comes from, or the bis. And the first and third carbon are phosphorylated, so that's where the 1,3 bisphosphoglycerate, that word comes from. So the ATP is used to phosphorylate the phosphoglycerate, and then NADH provides some electrons, and that converts the bisphosphoglycerate into glyceraldehyde 3-phosphate. All right, and so you've got 3 of these, 3 glyceraldehyde, you've got 6 of these, 6 glyceraldehyde 3-phosphates. 5 of these are going to stay in the cycle and be recycled or regenerated back into RUBP. 1 out of every 6 is the product. So this makes sense. You put in 3 carbon dioxides and you get 1 3-carbon molecule as an output. So with 3 cycles of the Calvin cycle, or 3 times we've combined RUBP with a carbon dioxide, and that has made 1 3-carbon molecule. If we wanted to make glucose, glucose has how many carbons? 6, people are saying. So if you wanted to make a 6-carbon molecule, you would need 2 G3Ps, and so you would need to put in 6 carbon dioxides. So you could also represent the Calvin cycle by putting in 6 carbon dioxides that are going to combine with 6 RUBPs, and that would generate 12 G3Ps, and then 2 out of those 12 could combine to make 1 glucose, and 10 out of the 12 would be regenerated to make RUBP. This is a complicated sequence of reactions that I am not going to even attempt to have you learn. But notice that you've got 5 3-carbon molecules, and as a result of combining and rearranging them, you get 3 5-carbon molecules. So you've got 15 carbons and you've got 15 carbons. So there's some rearrangement required, and there's more ATP required to do that. All right. Big picture is we're taking carbon dioxide out of the air, and we are building that carbon dioxide into organic molecules, which are the output. And that's called carbon fixation, fixing inorganic carbon to make organic carbon. And that requires ATPs and NADPHs. Alrighty then, I have another question for you. So that question is, if you want to synthesize 1 glucose molecule, how many molecules of CO2 would that require? Okay, so most of you are saying 6. A few said 3, but 3 carbons is only going to give you a G3P. And some of you just made other guesses, but 6 is the correct answer. If you want to get a 6-carbon glucose molecule, you have to have 6 CO2s. Let me go back to this figure again. I don't know, can you guys read this up here that says CO2? 3 CO2s. So if you put in 3 CO2s, you're going to get 1 3-carbon molecule. To make a glucose, you would have to have 2 3-carbon molecules to combine, so you'd have to have 6 CO2s come in. Now, how many molecules of ATP and NADPH are used to make 1 molecule of glucose? Let me show you the figure again, because I don't expect you to memorize it. How many ATPs and NADPHs to make 1 glucose? Okay, I've received 3 answers that are all incorrect. So far, still waiting for a correct response. Nope, still waiting. Oh, there, I got one. Okay, so Callista, can you explain to us how you got your answer?
I got my answer mainly from looking at the graph where you can see that there are 6 ATP on one side, and then there's another 3 that come up on the phase 3, and then 6 NADPH are used in phase 2 of reduction.
Okay, but your answer wasn't 9 and 6, was it?
Yep.
Oh yeah, it was. Trent, how did you get your answer?
It was just the same thing that you brought up earlier about how one go around would make 1 G3P for the output. So then if it's a glucose molecule, you just times 2 for everything.
Okay, and your answer was still not completely correct, because I think you said 12 ATPs and 12 NADPHs. Michelle, I think, has it right. Michelle said 18 ATPs and 12 NADPHs. So you have to have 9 ATPs times 2, because you want glucose. You don't want just G3P. So it's going to take 9 ATPs and it's going to take 12 NADPHs. This difference in the amount of ATP and NADPH is what explains cyclic electron flow, which is what we are going to next. So before we go into that, do you guys have questions about the Calvin cycle?
Just confirmation again. So you said that for how much NADPH and ATP for 1 glucose molecule, that'd be 12 NADPH and 18 ATP?
Correct.
Okay. Oh, that's what I wrote too. Okay. I was just making sure.
Okay, I maybe just missed it.
That's okay. That's okay. Thanks.
Yeah. Okay. Other questions? All right. So it's going to take 6 go-rounds of the Calvin cycle to make a glucose, which we typically think of as the product of photosynthesis. All right, so let's take a couple of minutes here to look at some alternative mechanisms of photosynthesis. First, we're going to look at cyclic electron flow, which is an alternative to the light reactions. And then we will look at C4 and CAM photosynthesis, which are alternatives to the dark reactions. And probably we'll just look at cyclic electron flow, and we'll look at C4 and CAM next week after I've given you a chance to read about them and do a reading assignment about them. So let's look at cyclic electron flow. In cyclic electron flow, instead of, well, let's just look at it, and I'm going to ask you guys to tell me how this electron flow differs from the linear electron flow that we've looked at. And you'll notice that kind of in gray, we're seeing the Z scheme. So we're seeing the linear in gray, and then you're seeing the cyclic electron flow in kind of the yellow that's more in the foreground of this picture. So send a private chat to your partner about how cyclic electron flow differs from linear electron flow. All right, let me just call on somebody. I don't know who I haven't heard from in a long time. Let me call on Sophie. Sophie, what's one difference that you guys came up with?
We decided, or we came up with that it doesn't make NADPH and that it only makes ATP and that the compounds are just recycled.
Okay, that's very good. So that is kind of the main point of cyclic electron flow, is that it does not make NADPH, but it does make ATP. So let's just remember back to, let me see, I'll go back to the Calvin cycle and remember that the Calvin cycle requires more ATPs than it does NADPHs. So plants do a little bit of cyclic electron flow to generate a little bit of extra ATP just to kind of balance out those numbers. Let me go back to this. Okay, so Sophie pointed out kind of the main thing is that cyclic electron flow does not make NADPH, but it does still make ATP. And she talked about recycling. So notice that the electrons just go in a cycle, and that is why it's called cyclic electron flow. The electrons are not coming from water. So water is not split and oxygen is not generated. And you can see that there is no final electron acceptor because the electrons just keep passing through all of these carriers. This still requires energy from light because in order for the electrons to go from this chlorophyll A up to the primary electron acceptor, that still requires light. So there's still light being accepted and there's still light being passed from pigment to pigment to pigment in photosystem 1. And that light is used to boost this electron. And then the electron goes to ferredoxin and then to the cytochrome and the plastocyanin and then back into the same chlorophyll. So the electron does not make its way into the NADPH, and that is why you're not making NADPH. So how do the inputs and outputs differ? We've kind of already covered that. The inputs are the same, the inputs are different because it requires light, but it does not require water. The outputs are different because it's making ATP, but it's not making NADPH. And again, making some extra ATP without making NADPH is a way to balance out the numbers that are needed for the Calvin cycle. And also, ATPs that are generated are used for other cellular work besides doing the Calvin cycle. And then plants also do cellular respiration because plants also have mitochondria. And so they generate ATP also by breaking down the glucose that they make in the Calvin cycle. Okay. I think we won't go into the alternative pathways, and I will post a reading assignment for you guys that'll be due on Monday, and you can read about the alternative pathways in the Calvin cycle, and then we'll start also getting into plant nutrition. So you guys have a great weekend. Stay on the meeting if you have any questions that you want me to address privately or a little bit more privately.